Okay, we are continuing our lecture about rate laws and finishing it with a conversation about catalysis. And I will try very hard not to get too excited about this conversation because catalysis is going to save the world, one could argue. It's at least helped us solve some major problems that we've faced as a society. So let's just start with the definition of what a catalyst is. It is a substance that will affect the rate of the reaction and actually the mechanism of the reaction, but it's not consumed itself. So this means when you're looking at a reaction mechanism, you'll see a product out of one of the steps or a reactant be used, but then reformed at the end of the reaction. So a great example of this, just to kind of get on our feet, is the reaction between ozone and monoatomic oxygen to form dioxygen. And we've got the mechanism shown here without a catalyst. And this is a very slow step. This is actually an elementary step of a larger reaction of the decomposition of ozone that we looked at in the previous video about mechanisms. Well, this step is very slow, but it can go much faster with a catalyst. And that catalyst, if we look at it, is going to be chlorine right here. So if we include monatomic chlorine with ozone, it will go through a two-step process. The first step is fast, and it will produce oxygen and this chlorine-oxygen compound. And then that reacts with another atom of oxygen and will form monoatomic oxygen and form dioxygen, and then that chlorine gas again. And so if we look at the overall reaction of this, we're going to see that O3 plus O forms O2, 2O2 like before, but we also have chlorine as a reactant and a product. And remember, when we were looking at summing up all of the elementary steps of our reaction mechanism to tell if it was reasonable, if we have something that is on the reactant side and the product side and it's the same, we can get rid of it. So the reaction can still be written just as O3 plus O forms 2O2. We just also have this catalyst that's present at the beginning and end of the reaction. Here's what this looks like in a reaction diagram. So our original reaction is this blue line right here, where our O3 plus a monoatomic oxygen will go through some transition state and then form two molecules of oxygen as our products. So our uncatalyzed pathway has an activation energy here that makes it a slow step, a very slow step. When we introduce a catalyst, we fundamentally change the reaction mechanism. This one-step process becomes a two-step process. And each step is going to have its own activation energy, as we saw when we looked at mechanisms. And the catalyst is changing this reaction diagram to reflect a different mechanism. But also our activation energy for these steps are significantly smaller than the activation energy of the uncatalyzed reaction. And that's how it speeds up a reaction, by changing the mechanism so the steps have lower activation energy barriers to form transition states. The analogy that I like to use for catalysts is that when you have a reaction, it's like people running around in random directions colliding with each other. But a catalyst will stand there and grab one reactant and the other and then bring them together. And it helps kind of like play matchmaker almost with our reactants along the way. So when we think about this in terms of the kinetic energy that our reactants need to have in order to form our products, our uncatalyzed reaction had a high activation energy, and so we had a smaller percentage of our collisions would have enough energy to overcome the activation state. And now that we have, or once you introduce a catalyst, then that activation energy is much smaller, right? And so there's going to be more collisions that take place that have enough kinetic energy to overcome our activation energy. So we actually see more molecules able to align themselves into the transition state for each number of attempts for these collisions. And so that's how the rate of our reaction increases. It's by actually lowering that activation energy. So there's two main types of catalysts, homogeneous and heterogeneous catalysts. Homogeneous catalysts are in the same phase as their reactants. They just kind of mixed into the solution. And the job of these reactants is they hold a reactant molecule in the proper orientation, basically. So that way, when a collision takes place, the orientation component of the whether or not the reaction will happen will be more likely to occur. And so it'll be floating around in solution, and usually it will react with one of our reactants first, and then another will collide, and then they're going to actually form our product. An example of this is sulfur and oxygen and water forming sulfuric acid. And so we've got a four-step mechanism here. This is fairly complicated, but you can see that after sulfur dioxide forms in the first step, if we introduce NO2 into this reaction, we will form an intermediate NO that will be able to be consumed in a reaction with just a little bit of oxygen, and will reform our NO2. So we get a net reaction that is going to have our overall reaction plus NO2 on the product and reactant side. And so NO2 is acting as our catalyst. And these are all in the gas phase, right? So they're all in the same phase. Oftentimes we see homogeneous catalysts in aqueous solutions as well. So you can dissolve a catalyst into your solution for it to be a homogeneous catalyst. Or you'll see them in the gas phase, most commonly. The other kind is what we call a heterogeneous catalyst, and it's going to be a catalyst that is a different phase than our reactant particles. The most common one of these is to have a solid catalyst that is inside a liquid or aqueous solution, or is exposed to a chemical reaction taking place in the gas phase. And our heterogeneous catalyst is going to react with one of our reactant molecules, and instead of just putting it in the right orientation, it's actually going to change that complex and make it more stable in its transition state, basically. It's going to kind of set up one reactant to be closer to the transition state, and that'll lower its activation energy. Let's take a look at what this might look like. So here's an example of a gas phase reaction that is conducted over an iron solid catalyst. And so we have hydrogen gas and nitrogen gas forming ammonium gas. So in this case, we have to break a triple bond on nitrogen and the single bond between hydrogen, and then reform 3 single bonds in ammonium. What happens is our gas particles actually come down and react with our iron catalyst, and they this allows us to break especially that nitrogen-nitrogen triple bond and have it interact or bond to the catalyst instead. And so that's how we're able to overcome a rather large barrier of breaking a triple bond much easier, in an easier fashion. And so then once these reactants are activated by our catalyst, then it's easier for our hydrogen and nitrogen to start forming bonds, like right here or right here. Now, once these bonds form, they'll keep forming. Now we're at NH2 on each of these. And then once that formation is complete and we have these NH3 molecules, they'll dissociate from the catalyst back into the gas phase. And so our catalyst, this solid, is basically absorbing gas, hydrogen, and nitrogen and releasing NH3 as a gas. Another type of catalyst that I'd like to call out that I think is more like a homogeneous, it's kind of in between the two, but it's homogeneous in my opinion just because it's dissolved in the solvent of your body. But proteins that catalyze biological reactions are called enzymes, and these are just catalysts. They're very large, very specialized catalysts, and they work by absorbing a substrate, like a reactant. It's a substrate would be the biology term, I guess, for it. A reactant into what we call an active site, which is a spot on a small spot on a very large enzyme that is designed to bind to the reactant. And then what happens is it actually holds it in a very specific orientation and sometimes also activates some of the bonds to resemble the transition state. And then it will release products. And in this case, we're looking at something where we have like an A going to a B plus C. But the same thing can happen where an enzyme, call this lock and key example, has two sites and it brings in two substrates. So this would be A plus B to form C. And then once they're in place, we've got the reaction that we're interested in having happen will be able to take place because it orients the molecules in the correct orientation to be able to form the transition state, and sometimes actually activates the bonds so that way, which means like partially bonding to them so that way they can more easily form that transition state. And these enzymes can actually, once they bind the reactants, they can actually change their shape a little bit so that way the molecules come together in that state much better, and then they can actually release it. And then we have an enzyme that can then go back to its original shape and bind more reactants. So this kind of goes in the decomposition way or in the forming of a new product from two different reactants way. So now I'd like to just go through a couple examples of this. So one of the reactions that at least bioinorganic and organometallic chemists are super interested in is the oxidation of methane to methanol. Methane is a gas, and methanol can be a liquid. So if you can imagine large deposits of methane gas in kind of remote areas, it'd be really useful to be able to convert them into a liquid and put them in a pipeline and ship them to a plant where you could use it as a chemical feedstock rather than petroleum. So we have it's thermodynamically favorable, which means to actually not do this reaction, but to actually do the reaction that goes even further, where you take CH4 and you actually react it all the way to carbon dioxide. And so the trick is to somehow stop this reaction at methanol without actually forming carbon dioxide. And that is actually the tricky part. And there are some specialized catalysts that have been designed to do this. A famous one is the Shavelev Cycle. It's actually a platinum catalyst. And so you have this catalyst right here is what it looks like. It's a platinum +2, and it's bonded to two water molecules and two chlorine ions. And our methane will approach this catalyst, and this will be a homogeneous catalyst, displace one of those chlorines and form a bond to the platinum. And what's happened here is the platinum is actually now activated this methane molecule. It broke a bond between it broke one of our CH4 bonds. And breaking one of these isn't exactly trivial. So it broke one of these and our hydrogen left. And now we have something that's able to react with something to form methanol, and that something is going to be a hydroxide ion. So this activated CH3, the platinum is going to change and become a platinum +4 to be able to facilitate a redox reaction. And so we have to have an oxidant around too, which is actually just more platinum. And then it'll reform this catalyst again when it releases methanol. And so these are the different steps of this mechanistic, or this mechanism, and we call it the mechanistic pathway. Another example of this is a copper zeolite catalyst. Now this is a very large structure you can see here. And what happens is we have our oxygen come in and bind at one of the sites within this larger nanostructure. And then we'll have methane come in and bind, and water will come in and bind. And from all of this, we'll release our methanol. And then we can keep binding these gases more and more and more, and then releasing more methanol on the side. And so this is something that is like a heterogeneous catalyst, but sometimes it can be particles small enough that they're actually suspended or even dissolved in solution if they're on the smaller side. Then, of course, so we've got we've looked at a homogeneous example, a heterogeneous example, and then, of course, there's the enzyme that does this. And there is an enzyme out there that catalyzes the reaction between methane and methanol, and it's an oxidation step. And so the oxidoreductase enzyme that we find actually in methanotrophic bacteria, which are really found in anaerobic environments, and they help cycle carbon. But this is something that's going to want to actually extract energy from methane. It will form it will undergo a reaction with methane that will produce methanol, and it's using this big old enzyme. Like, look how big this is. It's so big that we're not actually going to draw out the atoms involved. Instead, we're going to show what they are with ribbons and curlicues to show what the larger structure of this enzyme looks like, or shape. But where all of the chemistry really occurs in this large thing is at a small active site. And that small active site has two irons that are joined together by oxygens, and this would be one of our amino acids. And they have some more stuff going on over here that's going to bind it into the enzyme. And this is where our chemistry will take place. We'll have this kind of activated structure right here with oxygen coming in, and we form these iron 4 centers that each have an oxygen binding them in a dimer. And then when we have this intermediate here and methane is added to it, we can see that the methane's going to be able to react with one of these oxygen and water, and it's going to release methanol. And then what happens, since this is an oxidation, the reductant is going to be the iron, and it's going to, or the reducing agent, the iron will accept 2 electrons and form iron +3. And we now have a water right here. So here's a chemical reaction that we can see happen in the homogeneous, with a homogeneous catalyst, a heterogeneous catalyst, and something that we actually see in nature with an enzyme. Okay, I'm going to give one more example of another tricky problem that nature has solved, and that and that we are also always trying to solve. And this issue is splitting water. Splitting water refers to taking water molecules and forming hydrogen gas and oxygen gas. it's an endothermic process, so it's got a reaction diagram that looks like this, where we have an enthalpy that's going to require heat or energy to be absorbed by our reactants in order to be able to form our products, which are going to be less stable than our reactants. So it has to overcome this activation energy, and then at the end of the day, it still needs to input energy to be able to make this reaction take place. So this can be done by just passing electricity through water, or the electrolysis of water. The problem with this is it's inefficient. It's not efficient enough that you could actually use this as a way to store energy. However, in nature, plants have this figured out. Photosystem 2 catalyzes the splitting of water. It takes water molecules and it forms oxygen, and it separates it from the hydrogen, which is amazing. I mean, this is how plants use sunlight and to convert water to oxygen and create energy. And this right here is an image of photosystem 2. This is this huge catalyst. And in this huge catalyst is a small active site that we believe looks like this. And the actual characterization of the active site, which is again a metalloenzyme, so these metal centers, was very elusive for a while. And the interesting thing about this is that this active site works so hard that it actually gets replaced often within the enzyme. So it's mostly manganese, but there's also a calcium. And again, you can see lots of oxygen are involved in this, binding the manganese together. And here's a proposed mechanism for what is happening inside photosystem 2. Again, this is something that's still currently being researched and understood. And it's that, and this is utilizing, again, this is embedded in a large protein. So these are amino acids that are part of the overall enzyme over here, tyrosines. And so we have our water molecules right here that are going to, or here, we'll go from here. When we bind oxygen here, it's binding our water. We're going to bind our water right here and release oxygen. That oxygen is coming from this peroxo dimer that was created between the two manganese centers. And so here's the oxygen that's going to be released. That's our product oxygen here. So it's actually our product oxygen that's released is coming from this piece right here. And the water molecules that are being inputted into this, here we have some of our water molecules coming in here to displace our oxygen, are right here, and they track through. And so eventually they actually create this species here that's going to turn into the oxygen, the peroxodimer that will be released as oxygen. And studying these mechanisms in a biological system is tricky at best. So X-ray crystallography has advanced tremendously. And so we sometimes can grow crystals of an enzyme that freezes them in place at different spots along our mechanistic pathway, our catalytic pathway. And so you actually can see snapshots of different intermediates. Now, if you're lucky. Otherwise, the other way we try to understand these types of mechanisms is that we try to build small catalysts that mimic the active site of the protein, and then we can study how they interact with the reactants and products and then imply that that's what's happening or something similar is happening in the enzyme. So here's how nature does it. And we usually just pass a current through water and we can do it, but it's very inefficient. So in order to increase that efficiency, the addition of a catalyst would be ideal. And one has been in like around 2009, 2010 to 2011. A really big deal was a catalyst that was introduced by a group that worked at room temperature, which was special in water, and helped catalyze the electrolysis of water such that the activation energy was much lower, and that increased the efficiency of the reaction. And it's actually a catalyst that is cobalt and HPO4 minus, two common, not overly expensive reagents. And that can actually be used when you pass a current through to produce hydrogen and oxygen gas far more efficiently, which you can see bubbling. And while it's a little bit, it ends a little bit high level, it starts really well. I recommend watching at least the first 10 or 15 minutes, 10 minutes of this talk that Daniel Nocera gave, who is the primary investigator of the group that reported this research about creating an artificial leaf. And here's their artificial leaf that they developed. And the goal of the research is really to take the energy that a solar cell absorbs, that creates, and find a way to adequately store that energy to be used later on, tackling the storage problem of solar and renewable energies rather than the other end of it, which is creating an extremely efficient photovoltaic cell. So I recommend giving this a watch, and I have it linked as well on this page. So at this point, I think you should practice some problems. Here is a set of problem set problems. Number 5 is related to the mechanism section of the lecture, and this is a brief one about homogeneous and heterogeneous catalysts. So recommend going and watching the video linked to get a better understanding of how we tackle problems in the chemistry industry through methods like catalysis. I'll post the detailed solutions to this as well, so you can compare. I'd also remind you that we have overview slides that are posted online that you can look at that review the entire chapter as we go into an exam next week, and review rate laws, collision theory, and mechanisms, and then catalysts, and the different types of catalysts.